You have a minor.
Fisher my least favorite bird.
I always get Fishers on
I mean, there's
mine.
least favorite what?
Yeah.
Vertebrate?
What? You look pretty similar. Isn't it just the size?
There's interesting
And,
stuff.
Yeah, Martin
Yes,
is
I do.
probably about this big.
That's not even a question. Snakes is like top 3 on my list.
Fishers, though,
Fishes
they.
are prolific. I caught a blind snake just the other day.
Yeah,
Pretty proud of it, my
Fishers,
first one.
I would say they're like.
Blind snake?
Huh?
Like
Underground.
actually blind?
Yeah, they live in ants.
Oh!
They're the same size of a gray fox.
Their tail. Aren't those the ones that kind of look like their heads and their
With
tails
tails.
are like shaped similarly?
Yeah.
Or
Yeah,
like-
their
It's
tails
only
are
like this
long.
big.
Okay, I have a
Yeah,
different
pretty
one.
small.
Thick.
Honestly, I didn't even dig for it. I was joking, there's probably 100 ant colonies in my lifetime looking for them. This one just crawled up on my porch.
Oh, they're ants?
Yeah,
Gray foxes are the smallest.
it's pretty gnarly. I thought it was a worm, and then I was like, wait, it's got eyes on it.
Maybe it's a red fox.
How long is it?
There's a video of a fisher fighting a gray fox.
Can you afford it?
I can afford it. Why did they even? Should be free at that point.
Did you tell them for $200, bro?
Foxes,
Two times
differences of interaction, mutual interaction.
Right.
It's like the western spotted skunk is called a specific gray
I'm
fox.
down to go around.
So I had a question about number 2. Call it sunfish. Alright, import it. So check normality, check homostaticity. Should be good. I think number 4 was the only one that was not. Sorry. Forgot. It happens every year. Happens every year.
I'm sure nobody got the people.
I have to keep remaking that data set. But anyways, I'm gonna go through like model simplification and stuff for this one. So sunfish data set. We've got species, we've got sex. We're trying to look at size, see if size differs. All right, so I'm gonna make my full model, and I've already loaded the car package. So we're trying to predict size using the categorical variable species and sex. All right, so we're gonna make that full model, and then we're gonna look at that ANOVA table, see what it looks like. And when we look at it, the only thing we look at is this interaction term first. So if the interaction term is not significant, then we proceed and make our main effects only model. We look at it, we remove the one that has the largest p-value that's not significant. All right, and we continue simplification until we find out maybe we don't have any significant variables in our model. All right, maybe everything is not significant. In this case, we have a significant interaction. So this is our simplified model. All right, but we're not done. We have to come up with a description or an explanation for what's going on. So I'm just going to kind of make a plot. Interaction plot. We'll do species, sex, and size. See what it looks like. All right, so what we're looking for is trying to describe the effect. So right now, we know that the lines are not parallel. All right, we know that they're not parallel, that we have a much sharper drop between the blue and green. We have a decrease when we get to long ear for one sex, but basically a flat line for long ear. And then for red-breasted, it looks like they're about the same. But what we need to do is kind of go through and demonstrate that, yeah, there is a difference between the sexes. Like right now, it looks like that females are larger than the males. And it looks like bluegills are larger than the rest. But we're going to have to kind of go through and say, is that true? Are there any differences between green, long ear, and red-breasted? And because there's a significant effect, we need to restrict it to just the comparison of the males and just the comparison of the females. So what we're going to do is make subset. All right, so sex is M and F. Oh, I'm going to use this graph. Because this graph gives us the actual labels. All right, so You're gonna make a subset of the males. All right, you're gonna make a subset of the females. And when I do that, we are controlling for sex. So at these, we're going to look for an effect of species. And then I'm also going, since this graph is up, I'm going to go ahead and make my subsets. Subset species. Copy and paste that. Actually, we're going to do this. BG. G. LE. Or LE. I guess I'm going to do BG here. Capital BG. This is long year. And then RB. Red breast. All right, so I've got all of those. Just going to go through and make all of them. They all look like they worked. So now we're going to do our ANOVAs. So we'll start with the species. So I'm going to do this all in one set. So species, within each species, we're comparing male and female. And I know that if I get a significant difference, I don't have to do any posthoc tests because it's just 2 levels. So instead of making individual models and then passing that model to the ANOVA, I'm just going to run it all into one line. So we are looking at size as a function of sex, where our data is going to be dat.bg. All right, and I am pretty certain that's going to work, so I am just going to copy, paste it for the 4 data points, for the 4 data sets. And we're going to go one by one. So yeah, for bluegill, we have a difference. For green, we have a difference. For long ears, we have a difference. And for red-breasted, we have a difference. So I would save all of those p-values because we would need them for our write-up. So here's our interaction plot. What we just showed is that at each individual species, these 2 lines are significantly different from each other. The females are significantly larger with every single species. So we've done that. All right. So now the next step is to control for the sexes and compare across the line. Do we have variation in size between the species? Maybe the only difference that we find is for the males. Maybe we find differences for both male and females. So now, I'm going to go ahead and make a new one here. Now what we're going to do is use male and female, and I am going to start off with mod.full. We're going to look at size as a function. Actually, this is just, we're going to probably end up mod.n for males. Size is a function of species, where data is dat.n. Make that model, look at the ANOVA table. All right, we get a difference in species. So now, since all of our assumptions are met, we can just go ahead and run our Tukey's. And what we can see is that long ear and green are not different, red-breasted and green are not different, and red-breasted and long ear are not different. So basically, bluegill is larger than all the rest. These are all similar, at least within the males. All right. So we've kind of stepped through that interpretation. Now we have to repeat this for the females. So change that to females. And I don't know if we need Tukey's yet. What's that?
Never mind, don't worry.
So if we do, ANOVA tells me that we do have a difference in size within the females, size of the species within the female fish. So we do have to look at that Tukey's table. And what we see is, again, bluegill is different from green and long-ear, but it's not different from red-breasted, and all other are not. So our bluegill up here are different from these two, but it's not different from red-breasted, and it's pretty close. Red-breasted numerically it's a little bit smaller, but statistically it doesn't seem to be a difference. All right. So at this point, we've kind of done the analysis. We had the interaction, then we had to take it to the subsets to control for each of the variables, and then we kind of took that next step. All right. Did you get this? Do you have any questions on it?
How important is it for actually plotting switch
Did you switch them?
like you had to say on the bottom?
How do they look?
Yeah, I thought that.
I don't know. That looks pretty darn good. Let's look at it. Take a look at it. This one. Let's put it this way. If you presented this in your defense, I would fail.
I don't think it's good, Cody.
I'm gonna say, I think it depends on what you really want to show, because you do see the difference between the females and the males. I mean, you see that very clear, and it's also very clear that bluegill's higher than the rest. I think either can work. I think it comes down to personal preference. It's an opinion. I think I would probably prefer the first one over this, just because if you publish, you can't really publish in color. Color prices are way too much unless it's an online only journal. So here you have to use different hashes, different dotted lines. You've got 4 dotted lines here versus just 2 lines for this one. So you can kind of get by with the big less clutter. And don't forget, we also need measures of dispersion around here, right? So this works, but we should have error bars to show how much variation we actually have. This graph, error bars would be fairly easy to put on here. On this other plot, yeah, you can put the error bars, but you're going to see massive overlap, especially in these. So in these and
Better to go with less lines.
yeah, usually less is less. All right. Does anyone have any questions? Any questions on the homework? Other homework? No questions on question 4?
Well, maybe there's no interaction at all, is what I thought.
Okay, no interaction. What about the main effects? Yeah. Yeah, so you're in this situation where it didn't matter, those variables that you thought were important, they're not. They're not. So, and that's possible. It's possible. All right. Let us continue.
Real Welshes, real.
No, no. No, not in R. Not R. Let's see here. Figure out where we we are. All right, so where did we leave off? What's that? Radishes.
Yeah.
Nutrients, water. All right, so actually, I think we left off with like the GVLMA, right? Checking the assumptions and stuff, right? And then we said we were going to stop at radishes because we're kind of getting to that next step, right?
Yeah.
All right. So just a quick review. Once you start adding more than 2 variables, checking of the assumptions can become tedious. You're looking at normality within every combination of variables. You're going to have some groups that might not exist. You're going to have some groups that have a sample size of maybe 3 or 4. You're going to have others that have 15 to 20. You're going to start running into some sample size issues, which you can still do it. All right, you can still do QQ plots. All right, you can still do your Brown-Forsyth or your Flickner-Colleen test, but why go through all that effort when we have alternative methods, including this GVLMA? All right, so global validation of linear model assumptions is pretty easy, and we can also make our model and look at the graphical assumptions. Both of them are going to be okay. Nice thing about the GVLMA is that we get a p-value. We look at that very top line, that global stat line, and we get a p-value, and we can report that to our reviewers. We can report that to our committee and say, yeah, p-value is greater than 0.05. Our assumptions are met. And typically, no one's going to have say anything. Maybe Dr. Skipper says, hey, what is this GVLMA? Explain it to me. But I think everyone else will see R and a slew of numbers and p-values, and their eyes will glaze over, and they'll say whatever. All right, which is a good thing, really. All right. Disadvantage: you may detect slight variations, and there is some tricks. You look at the global stat line. Don't pay attention to some of the things underneath it, because if that global stat line is greater than 0.05, we stop. Our assumptions are met. Don't get caught in the trap that says, well, the global stat line p-value is 0.08, but our skew is 0.01, so we must be skewed. No, look at our global stat line. Plot of the residuals also works great. And the residual plot works for all different types of models. So, you can make your full model, look at the residuals, and go with it. All right, again, pretty easy to do in R. You just do plot of your model, and it makes those 4 plots for you. You can find code online that'll show you how to make each individual graph yourself using the residuals, which if you're looking for a larger version of the graph, that could work. But disadvantage, you don't have a p-value. All right, you're basically looking at the visual depiction of those residuals and making an assessment, making a subjective assessment as to whether or not those residuals are normally distributed, all right, if they're random and so forth. Do I have a preference one over the other? Not really. I usually check both, just kind of confirm it. Typically, I might start with the plot of the full model and see what it looks like and make a claim, and then see does GVLMA support that. So what we're going to do is move on to a 3-way ANOVA. And our practice problem is radishes. All right, so I'm going to want you to download and load that into R so we can work through it. All right, so with this example data set, we grew radishes under various conditions, and then we measured the diameter after 4 weeks. So that would be about the time when the radishes are getting ready for harvest. All right, so what we did with these various conditions is we altered nitrogen levels. All right, so in terms of the fertilizer, we varied how much nitrogen we gave to the plant. We altered how much water was being given to these radishes. And we had 2 different varieties. We had a regular growth variety, and we had a variety that's called fast grow, which is supposed to grow larger more quickly. that should reduce the amount of time from planting to harvest. All right? So I have these questions here because I want you to be able to import the data and then kind of figure out how all of this information was coded into the data set. So look at it, and then we'll talk about the coding parts, okay?
What package was Minecraft in?
That was
Probably
Oh, that was on.
Yeah, you have a couple lecture on it.
Oh, yeah, I got it.
Yeah, let's do that.
So, did you get it in? So what, for nitrogen, what were the treatments? High, low, medium. What about water?
Every day, Thursday, Wednesday, Thursday.
Okay. What about fast grow?
Oh, yes.
Okay, so it's either fast grow variety or not. What about diameter? And how'd you figure those out?
Down on
You did. I also just did summary. That works. As long as you don't have too many groups, this works. Now it does tell me some things. So nitrogen, high, low, and medium. It's alphabetical. I think as we do these analyses, we probably want to put them in a different order, either low, medium, high, or high, medium, and low. Put them in some sort of ordinal relation. All right. Water, I would probably do the same thing. So we have an everyday, an alternate day, every 3 day, and then a random. All right. Why random? Why not? All right. Why not? Someone says, well, how do you know that that you get improvement with every other day as opposed to just, it's getting water? All right. And then faster, I think no and yes. So go ahead and refactor, get things in proper order so that when we run our analysis, it should work properly. All right. Hopefully, you are comfortable in refactoring. So I refactored nitrogen and water. So when I look at my summary, I got low, medium, high. I get every day, alternate day, every 3 days, then random. And I didn't touch the fast growing, so left it as no index. That works. All right, so our interest in this data set is, does any of these 3 factors affect the diameter of that radish after 4 weeks of planting? All right, so one of the first things we need to do is look at our data set. Now, we made it to 3 different variables. So when we had, I should say 4 variables. When we had 3 variables, we had 2 categorical and 1 continuous variable, we can use the interaction plot very easily. When we add another independent variable, we can't plot, or we can't make a plot that has all of the variables together unless we go for some sort of 3-dimensional plot, which we could do. Could try that. But I'm not gonna spend an entire day showing you how to make 3-dimensional plots. Right? R, you can do it. And with R, you can also have it so you can move it and adjust it and kind of get it in the perspective that you want, but then copy and paste it, put it in your page. So there's a lot of power that you can do it. But here's one, nitrogen, fast growth, diameter. All right, so it looks like fast grow plants, the no's were much higher than the yeses in terms of mean diameter, right? But it's kind of hard to tell about the low, medium, high. Maybe there's not a difference. I mean, these look pretty flat. What about nitrogen and water? All right, I'm seeing something here. I'm seeing something where the lines don't seem to be parallel. All right, we have some crossing, so I'm not going to be surprised if we get a significant interaction between these 2 variables. And our last interaction plot is between the water and the fast grow. So what we've done is made 3 interaction plots to really investigate the effect. So again, looks like we've got differences in fast grow. It looks like we've got differences in our watering regime. But at least for this interaction, I'm not going to expect a significant interaction. The pattern seems to be the same. All right? So before we get to analyzing our data, we have to check our assumptions, right? All right? So go ahead, check your assumptions, and go through this process of simplifying our models to find the simplest model. Now, everything that we learned in that 2-way ANOVA applies. It's just we've added additional variables. We've added another variable. We just made it a little bit more complex. It depends on what our results are. Let me know when you get to check, when you finish checking the assumptions and after you look at the ANOVA of that full model. Can I see that jump where you're in the internet? These? So Cody had asked, are these 3 interaction plots required for the write-up? And my answer is, it depends on our model, on our simplified model, what our simplified model is. If there's a significant interaction, you're going to probably use the interaction plot that shows the interaction. But if it is simply main effects only, you can get by with box plots just for that. So for nitrogen as a function, or diameter is a function of nitrogen, and diameter is a function of fast growth, diameter is a function of water. You can get by with that. So which figures you present in your write-up just kind of depends on the simplified model. Did someone say they needed help? Oops, sorry.
Yeah, I do.
For which part?
So I think in my factory, and that's somehow changed my data in water.
Okay.
To change everything to NA. Don't know how to.
Oh, okay.
I have this.
Oh, because it's medium.
I made a full layer model and I did manual and I did Monday, so.
And then run that and see if it's. Okay, so do you have the chunk that reads it in? It's just reread it in. Because I think what happened was it just read that radishes line.
So you just make your model.
Scroll down.
Yeah, make your model and
Run that chunk again. There you go. All right, so did you check your assumptions? Yes? Almost? Great, as well. We'll talk, we're gonna do the assumption and then make your full model and look at that ANOVA table. And then we're gonna stop there and I'm gonna kind of give you suggestions on what we do from there. All right, did you get it? So I went ahead and made my full model, and then first thing I did was did the plot of mod.full to look at these residuals. So again, residuals versus fitted should be random. We appear in these groups because we have categorical variables as our independent variables. That's why it appears as it is. So for us being random, we're looking at approximately equal number of points and approximate equal number of spread. I should say approximate equal spread across each of our groups across this line. So we're not seeing this trumpet shaped pattern. We're not seeing a pattern that looks curved in any way. This looks pretty good. QQ plot of our residuals looks good. Scale location, again, we're looking at this line to be flat. This looks relatively flat. Not a huge change. We're between, let's say, 0.67 and 0.8. Not a huge change. And then our residual, again, yeah, we've got some labeled points, but we don't have those dashed lines, those cook distances on this graph. So that looks pretty good. I can use GVLMA on that full model just to confirm. And you'll see that you've got a huge amount of output, but I'm looking at this global stat line, which has a p-value of 0.65. So all of our assumptions are met. So what we need to do now is look at the ANOVA of that full table. All right, ANOVA of our full table, and what we're looking at is only this three-way interaction. That's the only value that we're looking at right now for this table. All right. All right, so we kind of covered this. All right, so first thing we're doing, this is basically doing like a stepwise regression. It's kind of how I learned it. It's probably not technically the correct term, but what we're doing is we're starting at a full model. We're looking at that three-way interaction. If it's significant, so if that three-way interaction is significant, then we have a lot of work cut out for us. Because what we're going to have to do now is start controlling for all of our different variables. So we're going to take our subset of fast grow. So let's say with all of our fast grow plants, then what we're going to have to do is do two-way ANOVAs. And if any of those two-way interactions are significant, then we're doing a subset within subsets to kind of do all of that analysis. If that three-way interaction is not significant, then we're going to drop it from our model and only look at our two-way interactions. So our question is, are the various two-way interactions, the three I think that we have here, are any of those three significant? All right? Any of them that are not significant, we want to drop from our model. And the process is to drop the one that has the largest p-value, the one that is least significant, so meaning the largest p-value. So drop it and then look back at your two-way interactions until you have a situation where none of the two-way interactions are significant, or you have only significant two-way interactions in our model. All right? So if we have only two-way interactions in our model, then we have to take subsets to interpret that interaction. If we have no two-way interactions, now we're on to our main effects. And we do the same thing. We remove the terms that are not significant. Our choices is to remove the one that has the highest p-value first, and then continue looking at our model. Now, you're going to see some changes in the p-value, because as we remove terms, we're moving that explained variance into our residual term, into our error term. So the p-values can change, but in general, from my experience and all this practice that we've done in these classes, if you look at three main effects and two of them are not significant, chances are, as you start removing terms, those two will be the two terms that you remove. And that applies to our two-way interactions and any other higher interaction term, generally speaking. But, so you can't wipe them all out all at once, because if you remove one, You're putting all that information in the residuals, and all of a sudden you could make one of those terms significant. All right? So how do we go about doing this? Because we can look here and say, well, I'm just going to change this instead of multiplication here, water times nitrogen times fast growth. I'm going to change these to pluses. Instead, I want water plus nitrogen plus fast growth. But if you do that, watch what happens. Whoa, hold on. Let me fix that. May actually change it to pluses this time. We only have the main effects. So with a two-way, we could do that. Three-way, we can't. With a three-way interaction, what we will have to do is manually remove each of the terms. Now, how we can do that is up to you. The hard way, is to do this. Whoops. Is to do your LM. We're doing diameter. All right, and then what we need is water times nitrogen plus water times fast growth plus nitrogen times fast growth. So go through and write every combination. Note I didn't put a three-way interaction term, but I have all of my two-way interactions filled. I don't suggest this because once you add 4 or 5 variables, it's hard to figure out and hit every single combination. The easier way, that's why I'm going to delete that. The easier way, look at that, is to use the update function. All right, so the update function takes a model and updates it based on the changes that we specify. All right, now the model part of this is going to be easy. We're working with mod.full. We're going to update that and we're going to save it as a new model. All right, so we're going to save it as a new model, just like this. The hard part is the change. Now the changes is in the format of a period, squiggly, period, and then something else. All right? So the period on the left says we want all the terms, all the same terms on the left-hand side of the equation as what's in the full model. The period to the right of that, so that's I have it in the form of left-hand side, LHS, and right-hand side of the equation with the tilde sign squiggly right in the middle. So if I have a period on the here, so period squiggly, that means give me everything that was already in the left-hand side of that model. All right. If I did period squiggly period, where now we have a period on the right-hand side, we want all the variables on the left-hand side to remain the same. We want all the variables on the right-hand side to remain the same. But we don't want that. We want all of those variables, and then what we could do is subtract out or add additional terms to our model. So a typical one that we're going to use is we're doing a reverse, I guess you could say reverse stepwise regression. We started at a full model and we keep stepping down. So what we're going to do is remove terms. So for us, we already have our ANOVA table open. This is our full two-way table, or ANOVA table. This is the term we want removed. So what we're going to do is update this mod.full, that full model. We want period squiggly period. We want the same terms on the left-hand side and the same terms on the right-hand side. But what we want to do is subtract out our three-way interaction term. How do I know what my three-way interaction term is? It is in our table. I write it exactly as it appears here. And what you could do is just highlight it, copy it, and then paste it. And now what I've done is I'm saving this updated model as mod.2way. So instead of a mod full with all of our three-way interactions, now I only have two-way interactions. I'm gonna run that and look at my ANOVA table, and voila, we have only two-way interactions in our model. So we've dropped out that three-way. Now we're at the two-way interactions. So what's that?
I got an error message says residual sum squares at 0 with the rounding error.
Okay.
Is it a
Squiggly.
Oh, that's
Filter.
okay. Period squiggly and then squiggly water.
No,
Or
no,
hyphen water.
no, period, squiggly, period, and then minus
Line, okay.
water, nitrogen, fast growth. Because, and again, if you change the order of this full model, so if you did nitrogen, water, and fast grow, it's going to be a different order for you. That's why it's probably easier just to copy and paste the term that you want to remove. All right. So we've looked at this. I'm looking at my ANOVA table. We are looking at only these two-way interactions. And we have two of them that are not significant. And we actually have three of them. Right? Now you might be tempted to say, well, let's remove all three of them, but we can't. What we have to do first is remove them one at a time, just to confirm that as we simplify our model, one of these terms doesn't all of a sudden become significant. And why is this? Well, again, this is an ANOVA, a type 2 ANOVA table. These two-way interactions, these p-values are the p-values after we account for everything in the model at the same or lower level. So this water fast grow interaction, this p-value is after we've accounted for water nitrogen fast grow and these other two interactions, the water nitrogen and the nitrogen fast grow. This water nitrogen is the p-value after this one, is p-value after we've accounted for those terms at a lower level and all of the rest two-way. If there's any sort of correlation between our variables, that can cause our p-values to change. It could change drastically. The example that I talk about is if we included month and if we included temperature. You have environmental water temperature in any of our studies. Water temperature is correlated with month. There could be a situation where month is not significant, where water temperature is not significant, but it's because we've accounted, well, after we account for month, temperature is not significant. After we account for temperature, month is not significant. You remove one of those terms, all of a sudden the other term becomes significant. That's an effective correlation between our independent variables. That's actually kind of an assumption when we start working more of this stuff. It's our collinearity assumption, but that's why. So work on these two-way interactions. We're going to update our model, removing a term one at a time, use the ANOVA table, and keep updating it until we get to a point where we only have main effects or we have a significant two-way interaction. Did you get it, Matt?
Yeah.
Right.
No, hold on.
We just have to remove each one of those, we don't have to replace it with anything.
Nope. We are removing our terms that are not significant. Let's go.
Should we remove nitrogen as well, or just stop?
How's it going? Probably. So, alright, we're here. We removed the three-way interaction. I used update. So we looked at this already. So our goal is to remove the least significant term. We use our p-value for that. So we're going to remove water fast grow. We look at our new ANOVA. And you can see how I'm naming them. All right, you don't have to name them this way. You're right, you can do mod 1, mod 2, mod 3, mod 4. But I would suggest, just from experience, if you keep like mod full, mod mains, mod two ways, and then just append some numbers after that, just to kind of keep track of your simplification process. So if you come back in a year, you're understanding what you were doing. So, you know, we remove the term, we look at nitrogen fast grow, that's not significant, so we're going to remove that. All right, we look at nitrogen water, that's not significant, so we remove that. All right, and now we've got nitrogen, water, and fast grow, nitrogen's not significant, so we remove it. And we go from this full three-way model with three-way interactions, two-way interactions, and so forth, and we're down to something rather simple, where water and fast grow is affecting the diameter of our radishes after 4 weeks of growth. All right, so what we've done is simplified it to get to the point where we only have significant terms in our models. Are we done? What else do we have to do? And
Well,
why do we have to do an ANOVA on the water and the fast growth? Because of what?
There's like levels within that
Well,
category.
because there's only 2 levels of fast growth. Well, no, never mind, I'm just talking.
Yeah.
You don't need to do a postdoc.
Yeah, so this is our ANOVA. This is it. We don't have to do subsets because we don't have any sort of interaction term. So at this point, we found our simplest model. Now we have to do our 2Ds. All right? But the trick is we can't just make a regular ANOVA with fast grow and a regular ANOVA with water because we need 2 keys to account for the other variable in the model before it figures out which terms are different. So the way we approach this then is to go ahead and take that simplest model, which is what I called it. So I did everything here. I got mod.nf, so for nutrients and fast grow. Just do 2 keys, HSD of the AOV of our simplified model, and 2 keys will do the rest for us. All right, it shows you fast grow, P adjustment is 0. 2 levels. All right, it's going to be simple. We just look at the figures, look at the averages of the fast grow. And that tells us which terms are, or it tells us which one is larger. For the water, we could see that every 3 days and every day had no effect. No difference. Random and alternate days had no difference. So let's kind of view this. Let's make 2 of these plots. All right, we don't need an interaction plot because we don't have a significant interaction. So what we could do is just do a plot like this, box plot, and say this is the effect of watering on the diameters. And you can see that every day and every 3 days had the lowest diameters. Alternate days and random days had the highest diameters, and there's no difference. So we can use our letters above these and say these are A, these are B, and say bars or boxes with the same letter are not different. For the fast grow, we could do this. These are the 2 figures that we could use as part of our write-up. What's that? Why would the random water have effect? I don't know. I needed a 4th group. So I made it random. So let's see here. Yeah, so every day you can rationalize it as being too much water, you get mold growth. Every 3 days, too much time passes between water, so they start drying out. Alternate days, that's optimum, but then the random is just as you're getting, again, maybe the random days is random between every day and every 3 days. So you're kind of in between. There it goes. Again, I made these things up. All right. So yeah, we do our 2 keys of our simplified model. And I did have in the presentation, and oftentimes what I do is convert that model name to mod.sim, just so I know that this is my simplest model. And then I do everything else with that simplified model. So here's a write-up. You can note here, so I did means of standard errors. So I used the aggregate function to get this. All right, to get those values. Made these plots. I made these in R, and I didn't. I don't think I gave you the code for that, but I could if you're interested. Note my figure legend. Diameter plus or minus SE to emphasize that my error bars are standard errors. Right. Important to add. So diameter of radishes, and then in A, figure A, diameter is a function of variety. B, diameter is a function of watering frequency. Values with similar letters are not significantly different. 2 keys HSD means that P is greater than 0.05. All right, so here in the write-up, you will notice I do report the global stat line for the GVLMA. All right, in the methods, I would have to give a citation to that GVLMA package. And then that's an R. R tells you, here's how to cite it. Here's how to cite it. All right. You'll also notice, actually, I give it to you right here. Pena and Slate, 2014. It's an R. All right. And then I said model simplification using a stepwise regression. And I didn't give p-values and f statistics for every single interaction term that gets removed. All right. That kind of starts to become redundant. Maybe you do, but I think it's sufficient to say non-significant terms were removed until the simplest model was obtained. The simplest model for these data included the main effects of watering frequency and variety. And for those terms then, I give my F statistic, degrees of freedom, and the p-value. All right. To me, I think this is sufficient. I've never really had anyone say otherwise to me. But know that if a reviewer says, oh, you have to give everything, then you give everything. All right? But with this stepwise regression, it should be sufficient to explain. If we don't have a significant p-value, we drop it just to get to the simplest model, because that is what we're looking for. All right? And then fast grow variety produces smaller radish, and the largest radishes were obtained with random watering or watering on alternate days, figure 1. All right. That's sufficient. Not terrible. All right. As part of this, as we start adding variables, all right, it's not, someone could ask you, well, how much an effect is there with fast grow versus watering frequency? Which one has more of an effect? This is where explained variance comes into play. All right, explained variance is basically the amount of variation in our diameters that's accounted for by our independent variable. The explained variation is quantified using the coefficient of determination. This is our R squared, typically capital R squared. Lowercase r squared tends to be a correlation coefficient. You're going to see it used interchangeably in papers, but I definitely recommend use capital R squared. It has a specific meaning. All right. So percentage of variation that's explained in our model is 0.8827, which means 88% of all of that variation in radish diameter is accounted for by radishes, by radish variety and watering frequency. Now, how did I get this? How did I find this term? How did I find this 88%? Where was it? Summary of what? Yep, summary of our simplest model. So, summary of, it should be mod.sim, but I didn't do that. Is summary mod.nf right here. Now, I would probably use this adjusted R squared, because the adjusted R squared is going to account for differences in sample size. But our simplest model accounts for 88% of our variation, which is a huge amount, right? Huge amount. But this doesn't tell us what percentage is accounted for by fast grow, which percentage, or what percentage is accounted for by our watering frequency. Is there a way to do that? Yeah, we'll have to do it by hand. So how did R do it? How did R calculate the R squared? Well, it took the sums of squares that is accounted for by all of the terms in that simplest model. And it divided it by the total sum of squares. Now, if you remember, total sum of squares is the sum of squares of each of our different variables, plus that unexplained variance, that sum of squares error. All right, so we can get it just right from our ANOVA table. So I'm going to roll back a little bit, right here. All right, we've got sum of squares here. We've got our residuals. Well, what's our total? Here it is. Bring my calculator over here so you can see it. All right, so we want to see this 88%. So it's 17,881. I'm going to use parentheses. 17,881. That's our water, sum of squares, plus, darn it. Let's clear this out. Let's try this again. I have equals instead. 17,881 plus our 59,398. Those are the two variables in our simplest model. And we're going to divide it by our total, which is 17,881 plus 59,398 plus 10,265, that error term. Close it, hit enter, I get 0.8827. This is the unadjusted R squared. Didn't account for sample size. All right, what about the unexplained variation for water? What about for fast grow? Well, it uses the same formula. It's the sum of squares over our total. All right, so for water, it's 17,881 divided by our total. So watering frequency accounts for 20% of that variation in diameter. Not super huge, but still significant. 20% of the variation is accounted for by diameter. What about fast grow? Well, we know already this is a higher number. That should be a higher percentage. What percentage is that? Any guesses? 67.85. Man, you are a human calculator, right?
Yeah.
Yep. Well, it should, because we've accounted for 88, right? 67.85. So 67.85 plus the other 20% gets us to that 88. So 88% of our total variation is accounted for by these two variables. Fast grow had accounted for most of that variation in size. Right? Why is this important? Well, as you work with more and more variables, it becomes important to start ranking which terms are the most important. Because if you have a decision, if your decision is to produce the largest radishes, what are you going to try to do? Should we focus on the strain of the radish, or should we focus on watering frequency? Well, based on this analysis, it seems like we should be focusing on variety. That's where we place our emphasis. Both of them are important, but fast grow accounts for the most variation. There's no easy way to do it other than doing it by hand, and this is where we account for it. So this is how we did it. And I just kind of showed you how I did it on the calculator to get all of these. You're probably looking at it.
Yeah.
All right. So we did the three-way. As we add more and more variables, it becomes the same. The process becomes the same. We start with a full model. So we check our assumptions first. If all of our assumptions are met, we start with this full model. We start with a full model. We look at all the interactions, and then we start removing one at a time those terms that are not significant. All right, so we always begin with the highest order interaction, and then we work backwards to find our simpler model. There's a catch, though. What if we have a significant interaction term? Then both of those main effect terms have to be in the model. So we can't go through, let me switch here. Let me move R over here. just for example here, let's say we have water and nitrogen. Let's say this was actually 0.045. All right, we get to the point where that was this one. Here it is. So we get to this point, we have a significant interaction. We can't then jump down and remove nitrogen, even though it's not significant. Nitrogen has to remain in the model because it has a two-way interaction. However, fast grow is not included. So if fast grow was not significant, it's okay for us to leave that two-way interaction and remove fast grow to get to our simplest model. So there are rules on how we're supposed to simplify. And you can see that doing it by hand, it can take some time. It can take some time to do it. But our goal is always to find that simplest model and then interpret that simplest model. All right? All right, so I do have a practice jalapenos. All right? Multi-interaction. So go through, work through this. On Wednesday, we'll kind of go through working through this, checking it. I'll probably go through it rather quickly, but go ahead and work through it as a practice problem. I do think it should, all of our assumptions should be met at this point. But you'll notice we did three-way interaction for radishes. Oops. We've got 4 variables here. All right, so we've stepped up to a 4-way ANOVA because, again, the process is the same. All right, let's call it. And I didn't go through all this, but you can use this as a guide. It comes back. All right. Let's hope we don't lose power and water again.
What happened to that?
I mean, it's bad.
What is an
It was about,
explosion?
a month or something completely dry.
Crazy.
You got a specific notification where Optimus Prime exploded, man.